Hi guys, today we'll talk about regulation of respiration. Regulation of respiration is both automatic as well as voluntary. We can blow a balloon voluntarily and without our notice we are breathing in and out. So it's both automatic as well as voluntary. And how here our concern is how it is automatically controlled. So there are three mechanisms which are involving in the automatic control. One is called as neural mechanism, another one is called as chemical mechanism, and the third one is the other, we are considering it as non-chemical mechanism. Coming to the neural mechanism, the voluntary part of the neural mechanism is under the control of cerebral cortex. So cerebral cortex is concerned with voluntary control. So if I am blowing a balloon, I can blow a balloon voluntarily. So my cerebral cortex orders the inspiratory muscles like diaphragm and the external intercostal muscles to blow the air out. And coming to the automatic, here we have four centers which are located in two regions, medulla and pons. So there are two medullary centers and two pontine centers. If you see here, this is the pons. So in the pons we have pneumotaxic center and apneistic center. So in the pons we have pneumotaxic and apneistic center. And in the medulla we have dorsal respiratory group and ventral respiratory group. Dorsal respiratory group here is concerned with inspiration and ventral respiratory group is concerned with expiration and inspiration. So medullary centers are concerned with generation of the respiratory pattern, whereas the pontine centers will regulate the medullary centers. Now we'll talk about the basic medullary centers. So in the medullary centers we have dorsal respiratory group. We already mentioned it is concerned with inspiration, and the dorsal respiratory group is located dorsally in the medulla. That's why we are named it as dorsal respiratory group. We will use the term DRG for dorsal respiratory group. And the neurons present in the dorsal respiratory group are of nucleus tractus solitarius. We name them as NTS. So the neurons in the dorsal respiratory group are nucleus tractus solitarius, and it is primarily inspiratory, and it also integrates the sensory information that is coming from glossopharyngeal as well as vagus nerve, 9 and 10 nerves. And as it is inspiratory, it gives the information to the inspiratory muscles like diaphragm. So the nerve is phrenic nerve, and the external intercostal muscles, so the external intercostal nerves. So dorsal respiratory group is located dorsal in medulla, and NTS, nucleus tractus solitarius, and it is inspiratory. Coming to the ventral respiratory group, it is located in the ventrolateral part of the medulla. That's why we call that as ventral respiratory group, VRG, and it is having nucleus ambiguous and nucleus retrofacialis, and it has neurons of both inspiratory as well as expiratory. So it acts both in inspiration and as well as expiration, but not under the normal conditions. So it is not active during normal inspiration or expiration. It is only active during hyperventilation as seen in a physical exercise. Now it has three regions: the rostral, middle and the caudal regions, and it is bilaterally present in medulla. So the other half is also having the similar part, and the rostral, middle and the caudal. Rostral and the caudal part is having expiratory neurons, and the middle part is having inspiratory neurons. And as we said, it is not active during normal inspiration. So in hyperventilation, it increases the caliber of the airways. And not only DRG and VRG, nearer to this DRG VRG complex, we have a central pattern generator. Central pattern generator is nothing but pre-Botzinger complex, which contains pacemaker cells. These are the ones which initiate the stimulus to the dorsal respiratory group of neurons, and it generates normal respiratory rhythm. So the inspiratory rhythm is also called as inspiratory ramp. So the signals coming from the pre-Botzinger complex are not single bursts. They will increase in their amplitudes. So up to 2 seconds, let us think that the amplitude of the stimuli is increasing, and suddenly they will cease and there occurs a pause for 3 seconds. So total of 5 seconds, that gives a rate of 12 breaths per minute. So 2 seconds, the inspiration will occur in a ramp like pattern. So the stimuli increases gradually and suddenly ceases. That causes the expiration. So what is the benefit of this inspiratory ramp? That causes the smooth expansion of the lung. So the lung won't expand in like inspiratory gasps. It don't expand like that. It helps in the smooth expansion. And after the 2 seconds, when the signal is ceased, the recoil of the lung causes the expiration. So expiration is simply passive recoil of the lung. If it is a voluntary expiration, maybe the internal intercostal muscles will come into the action. And other than the central pattern generator, there is an integrator or control which controls the activity of central pattern generator. So how it controls? It gets the information from the cortex, limbic system, mechanoreceptors, chemoreceptors, and even from the receptors of lung and chest wall. So cortex, the voluntary information; limbic system, the emotional information; proprioceptors from the joints during the exercise, and chemoreceptors which are present in the carotid body, and the mechanoreceptors which are present in the chest wall. And this helps in the adjustment of rate and depth of the breathing, and also it controls the energy expenditure. It reduces the energy expenditure. Now coming to the Pontine respiratory centers. We have two Pontine respiratory centers, pneumotaxic center and apneustic center. So pneumotaxic center contains neurons of nucleus parabrachialis, and it is active during both inspiration and expiration. And what is the role of this pneumotaxic center is it inhibits apneustic center. Whenever apneustic center is inhibited, apneustic center is concerned with inspiration. So inspiration is cut. So it ends the inspiration and begins the expiration. So it switches between inspiration and expiration. Coming to the apneustic center, it is present in the caudal pons, and it activates medullary inspiratory center, that is nothing but the dorsal respiratory group, and it is inhibited by the pneumotaxic center. And the stimulus causes prolonged inspiration, that is called as apneusis. And not only these four centers, we need a sensory information from the lung, how much is stretch. So such an information is carried by the vagus. It carries the information from the lung and airways, and what it senses the inflation of the lungs, that is stretch of the lungs, and it inhibits the inspiration. So it cuts the inspiration. Okay, enough, the lung is stretching more, you just stop, so that inspiration is cut off. And vagotomy, so there is no sensory afferent, and the lesion of apneustic center causes apneusis, which is prolonged inspiration. And the higher centers, we already mentioned, cerebral cortex is concerned with voluntary control, and the limbic system is concerned with the increased respiratory pattern during emotions, and the reticular activating system increases the respiratory rate whenever we are awakening. Now coming to the overview of this neural mechanism. So we have a pneumotaxic center, apneustic center, dorsal respiratory group, ventral respiratory group. So this is pneumotaxic center, apneustic center, dorsal respiratory group, ventral respiratory. Now, this dorsal respiratory group is concerned with expansion of the lung. So, it stimulates the inspiration. And this apneustic center also facilitates the action of dorsal respiratory. So, both apneustic center and dorsal respiratory group are stimulatory for inspiration. Now, the pneumotaxic center is inhibitory over apneustic center. That means apneustic center no more can stimulate the dorsal respiratory group. So, if pneumotaxic center is activated, inspiration is cut. If apneustic center is activated, inspiration is favored. Now, the information from the vagal efferents stimulates the pneumotaxic center. Now, apneustic center is cut. So, inspiration is cut. So, this is how the neural mechanism acts with the help of Pontine centers, medullary centers and vagal efferent. And what happens if there is a transection? Or to know the function of this, we make a lesion. So, let us think that this is the pons. I made a lesion above the pons. So, above the pons means maybe the voluntary control by the cortex is lost, but the respiratory rhythm is maintained by the medulla. So, that is intact, and it is controlled by pneumotaxic center, that is pons, that is also intact. So, whenever the afferent from the vagus, so vagal afferent is intact, the respiratory pattern won't change if the lesion is above the pons. But when the vaga is cut, the frequency of the breathing pattern is reduced and the depth of the respiration has increased because stretch afferent is cut. So, lung is expanding more and the frequency is reduced. Now we are making the lesion at pneumotaxic center, below the pneumotaxic center. That means apneustic center is intact. So, if the apneustic center is intact, what happens? You can see the depth of the respiration is increased, but the frequency is reduced because pneumotaxic center is the one which is essential for frequency. And now the vaga is cut. You can see there is a prolonged inspiration. We are calling that as apneusis. Now the lesion is made below the apneustic center. That means medulla is now independent of pons. So, the depth of the respiration is increased. So, switching between inspiration and expiration is cut. And when the vagae are intact, okay, the pattern of respiration is maintained, but the depth is more. We are calling that as gasping. But when the vagae are cut, you can see that the respiratory pattern becomes irregular and cessation occurring. So, gasping occurs. So, irregular pattern occurs if only medulla is intact and the Pontine connections are cut. And if the lesion is below the medulla, the person dies of respiratory failure. There will be no respiration. So, with this we can know that pneumotaxic center is concerned with frequency and apneustic center is concerned with depth and medulla is concerned with a rhythm, but under the regulation of pons. And below the medulla there will be no respiration. Coming to the chemical control of breathing, it is controlled by the levels of oxygen, so decreased partial pressure of oxygen, hypoxia, and carbon dioxide, increased partial pressure of carbon dioxide, that is hypercapnia, and increased H plus acidosis. So, hypoxia, hypercapnia and acidosis are the stimuli for the peripheral chemoreceptors as well as central chemoreceptors. So, we have peripheral chemoreceptors which are carotid body and aortic body. Carotid body is carried by, the information from carotid body is carried by 9th cranial nerve and the aortic body is carried by 10th cranial nerve. And coming to the carotid body, where carotid body is located? At the bifurcation of the common carotid artery. If you see the aortic body, which is located in arch of aorta and ascending aorta, and carotid body is carried by glossopharyngeal nerve, which is 9th cranial nerve, and aortic body is carried by vagus nerve, which is 10th cranial nerve, and they gives that information to the respiratory centers in medulla. Carotid bodies are located at the bifurcation of the common carotid artery, and usually they weigh around 2 grams, and it has the highest blood flow based on 100 grams of the tissue. So per gram weight, highest blood flow is to the carotid body, which is 2 liters per 100 grams of tissue per minute, and which is almost 40 times the blood flow to the brain. Why this tiniest body requires that much blood flow? Because it has to detect the changes. So it can easily detect the chemical changes, and even it has the high metabolic rate, so utilization will also be more, and that can be easily detected. And there are two kinds of cells in the carotid body. One is type 1, which is also called glomus cells. This is the receptor for these chemical changes, and it is neuroectodermal in origin, and it contains voltage gated calcium channels. Let us think that this is the glomus cell, and these are the dopamine vesicles or neurotransmitter vesicles, and the surface contains calcium gated voltage channels, and it releases dopamine whenever it gets excited, and this dopamine will act over the D2 receptor, which is present on the 9th and 10th axons, and it is surrounded by fenestrated capillaries which have pores in their walls, so that it can easily detect those changes. And the other cells are type 2 cells, which are the supporting cells. Aortic bodies, as we said, present in the ascending aorta and arch of aorta. Coming to the mechanism of action, whenever the oxygen content is lowered, that causes closure of potassium channel. So K plus is closed. So K plus can't go out. So K plus accumulates here, and that causes depolarization, and that causes opening of voltage gated channels, which are calcium, and the calcium influx occurs. Exocytosis of neurotransmitter, mainly the dopamine, occurs, which acts over D2 receptor, generates an action potential in the axon. And if you see, whenever the oxygen is lowered, the firing rate from the carotid body is increased. So to get depolarization, the prime thing to occur is closure of K plus channels. Coming to the three factors, in that first we'll talk about hypoxia. Hypoxia is the potent stimulant and the main stimulant for the peripheral chemoreceptors, and PO2 of less than 60 mmHg, the firing rate increases rapidly. If you see here, arterial oxygen saturation is getting lowered, but the firing rate or the ventilation is not much increased, up to 60. After less than 60, you can see that ventilation rate almost increased 6 times rapidly, and PCO2 is kept constant at 40 mmHg. So hypoxia causes hyperventilation, which causes washing out of CO2. If CO2 is washed out, that inhibits breathing. So respiration is inhibited. But hypoxia, to overcome this inhibitory effect of decreased CO2, the PO2 should reduce less than 60 mmHg. Until that, the CO2 should be kept constant to cause the stimulatory effect of ventilation by PO2. How CO2 is kept constant? By having oxygen carbon dioxide mixture. Coming to the how hypoxia, there are three probable mechanisms which causes the closure of potassium channel. One is there is a protein which is heme which is bound to oxygen. So whenever oxygen from heme containing protein is lowered, closure of potassium channels occur. This causes decreased potassium efflux. So K plus remains inside and that causes depolarization. So one way, or it inhibits the cyclic AMP mediated potassium channels by increasing the cyclic AMP. So K plus closure again, or it inhibits mitochondrial NADPH oxidase, which again inhibits the potassium channels. So potassium channels can be inhibited by one heme oxygen is less bound to heme containing protein, which causes K plus closure, or increased cyclic AMP, or decreased NADPH oxidase of mitochondria. These are the ways which hypoxia can cause closure of potassium channel. Coming to the hypercapnia, if you see the hypercapnia, whenever the CO2 levels are more than 40, you can see that there is a linear increase in the ventilation rate. Even the pH, whenever it is lowered, the ventilation rate is increased. So hypercapnia has a linear relation, and hypercapnia, how it is causing the closure of potassium is H2O, carbon dioxide enters the glomerulus cell, binds with the H2O to form H2CO3, which is carbonic acid, which is weak, unstable, and it dissociates into H plus and HCO3 minus. This H plus will block the potassium channel. So the same, decreased reflux, depolarization. Acidosis. Acidosis is also acting similarly, but it inhibits sodium H plus exchanger. So H plus accumulates in the cell. So that inhibits the K plus channel, again depolarization. Coming to the central chemoreceptors, they are more sensitive to H plus, but this H plus won't cross blood-brain barrier. So CO2 plays a role here. CO2 binds with water to form carbonic acid, which dissociates into H plus and bicarbonate. This H plus will act over central chemoreceptors which are present in the medulla, and that increases ventilation. So they are located in medulla and they change H plus, but H plus is less permeable at blood-brain barrier, but CO2 is permeable. So this H plus will cause the increase in ventilation by stimulating the chemoreceptors directly. Non-chemical mechanism, integrated responses to hypoxia, hypercapnia, acidosis. Let us think that PO2 is lowered, so peripheral chemoreceptors are activated, and this causes increased ventilation, and that causes washing out CO2, so decreased CO2 finally inhibits the ventilation. So hypoxia alone can increase the ventilation, but it is inhibited until it goes beyond PO2 less than 60 mmHg. Whereas hypercapnia plus hypoxia will cause marked increase in ventilation. So stronger in presence of hypoxia. But decreased CO2 will inhibit ventilation even there is hypoxia. Similarly, increased pH decreases the response to hypoxia. So hypoxia is the stimulant at peripheral chemoreceptors, whereas carbon dioxide and H plus are the stimulants at the central chemoreceptors. Even though hypoxia is the stimulant, but if CO2 or H plus are lowered, it can't stimulate the ventilation. Non-chemical mechanisms, where there are three kinds of receptors: slowly adapting receptors, rapidly adapting receptors, and C fibers. So Herring-Brewer reflex is a slowly adapting receptors where they respond to airway stretch or increased airway transmural pressure, which the response is they inhibit inspiration and prolongs expiration. There are two kinds of reflexes: inflation reflex, deflation reflex. Herring-Brewer's inflation reflex is the one where when there is an overstretch of the lung, like which occurs at tidal volumes more than 1.5 liter, so more stretch of the lung, it prevents overinflation. So vagus will be stimulated, that information goes to the brain and that stops the respiration. So it usually operates tidal volumes more than 1.5 liters, prevents overexpansion of the lungs, whereas deflation reflex, marked deflation, if you are exhaling the air out more, it decreases the duration of expiration. That is called as Herring-Brewer's deflation reflex. Rapidly adapting receptors, they are present in larger conducting airways. They adapt rapidly and they respond to variety of stimuli like inflation, sudden stretch, chemicals like serotonin, histamine, bradykinin, prostaglandins, or irritants like smoke and dust, or inflammatory signs like congestion and edema. All these will activate these rapidly adapting receptors and the response will be coughing, gasping and prolonged inspiration. So if you stimulate these rapidly adapting receptors by any irritants, it will cause coughing, gasping and prolonged inspiration. Coming to the type C fibers which are unmyelinated, there are two pulmonary C fibers also called as J receptors and bronchial C fibers, and they respond to stretch, that is inflation, injury to the lung and congestion, inflammation and chemicals. Bronchial C fibers are more responsive to chemicals than pulmonary C fibers. And there is a reflex called as J reflex where the stimulation of these J receptors either by congestion or edema or chemicals will cause rapid shallow breathing, bronchoconstriction, increased bronchial secretions and cardiovascular depression such as decreased BP, hypotension, and even there is a loss of skeletal muscle tone. This is called as J reflex. Activation of pulmonary C fibers causes cardiorespiratory inhibition, bronchoconstriction and loss of skeletal muscle tone. Thank you for your patient listening.